Thanks everybody for joining us for the August installment of the monthly seminar on physical genomics. My name is Benjamin Keene. I'm a senior program administrator here at CPGE at Northwestern. Sorry, got a baby with me today, so sorry if there's a little noise or distraction. We're really excited to welcome our invited speaker today, Dr. Geeta Narlikar. Dr. Narlikar is the Kuo Professor and Chair of Biochemistry and Biophysics at University of California, San Francisco. Dr. Narlikar's group studies how the folding and compartmentalization of our genome is regulated to generate the many cell types that make up our body. Her laboratories pioneered the application of sophisticated biophysical approaches to study the mechanisms of macromolecules that regulate genome organization. Among many other honors, Dr. Narlikar's scientific work has been recognized by election to the National Academy of Sciences in 2021 and the American Academy of Arts and Sciences in 2024. Dr. Narlikar, thank you for taking the time to share your work with us today. For our audience, there will be time to ask questions at the conclusion of the talk, or if you want to type them into the chat box at any time during the talk, we will just address them at the end. So feel free to type those in as they occur to you. If you'd like to learn more about the research and training programs at CPGE, you can visit us at physicalgenomics.northwestern.edu or follow us on YouTube, Twitter, or Instagram. At this point, Dr. Narlikar, if you'd like to go ahead and start your presentation, I can turn the floor over to you. Thank you so much again for joining us. Really appreciate it.
Thank you so much for the really kind introduction and for the invitation to speak here. I'll get started and see whether, let me see. Okay, can you all hear me? I'm assuming you can.
Sounds good, yes.
Right, okay. And so today I'll share some of our published work and some of our unpublished work in the broad area of chromatin regulation. Let's see if I can move this forward. There we go. Okay. As everybody in this audience knows, but it's just good to kind of step back and reflect. When you package eukaryotic genomes into chromatin, it allows for functional compartmentalization. And so at a very, very simplistic level, you can have DNA shown in this very simple cartoon that is less compacted, and genes in these kinds of regions are more off and on. In contrast, you can have DNA that is more compacted, and genes in these kinds of regions are more off and off. And this kind of differential genome packaging allows different cell types to express different genes and adopt different cell identities. And it's not hard to imagine that if you mess up this process, you can cause a cell to have an identity crisis. And the best example of cells having identity crisis are cancer. A cancer cell does not know any longer what it's supposed to be. Okay. Now, this kind of compartmentalization is not static. And this is also something everybody in this audience knows. And it's constantly changing between genes being in open states that are more accessible and genes being in closed states that are less accessible as the cell responds to external stimuli or as a cell differentiates. And a major set of proteins and enzymes that promote these kinds of interconversions between less compact and more compact states of the genome are ATP-dependent chromatin remodeling enzymes or complexes. And so these ATP-dependent remodeling complexes can take chromatin, and which we'll come to in a minute, that is more open and convert it to chromatin that's less open and vice versa. Now, this is a very simplistic diagram, but as I'm sure many people in this audience also know, that this is really not how the packaged genome looks. It is much more crowded in a nucleus. So if you go deep into imagining how crowded a nucleus is, you can use these beautiful diagrams from David Goodsell, and you can begin imagining how chromatin remodeling enzymes might actually have to act in a highly crowded environment. So this is just a rendition, but this gives you a picture of how crowded genome packaging can be in a nucleus. Now, this is how it might look if you went into a nucleus, but if you then went further down a few scales, you can see that the smallest unit of chromatin or the genome packaging is a nucleosome. And so this kind of nucleosome is considered the basic unit, but people have also found broken nucleosomes, so nucleosomes that are missing parts of the packaging proteins of the histones. And so the questions that our lab has become interested in, and these are some of the questions that we're working on right now, is how does regulation happen at the nucleosome scale, and as well as the subnucleosome scale? And how does regulation at the nucleosome scale impact chromatin at the genome scale? And specifically, what happens when you start doing biochemistry in a crowded chromatin environment? And so what is our approach? We are unapologetic biochemists and biophysicists. What we try to do is to uncover the biophysical basis and consequences of nucleosome remodeling in different chromatin contexts. And then we let the data take us to new conceptual territories. Okay. And so with that lead in, today I'll share two stories. In the first part, I will talk about a new activity of a chromatin remodeler that we've discovered over the last few years. And this chromatin remodeler we found prefers to act on hexasomes, which are incomplete nucleosomes rather than nucleosomes. And the second part, I'll talk about our foray into trying to do biochemistry in crowded chromatin environments. So before I get into the data, I wanted to kind of just introduce folks to a few different views of a nucleosome. So shown here is two views from the beautiful crystal structure originally determined by Caroline Luger and Tim Richmond. And what you have is about 150 base pairs of DNA wrapped around an octamer of histone proteins. And here are two different views of a nucleosome. And the nucleosome is wrapped around a histone octamer, and the histone octamer has at its core two histones, H3 and H4, that form a tetramer. And you can think of two other histones, H2 and H2B, forming caps on either side of the histone tetramer. And so this is the cartoon version of a nucleosome. And so a nucleosome is wrapped around, is formed by wrapping DNA around a histone octamer, and you can think of the histones as being modular. Now, we know a lot about chromatin remodeling activities from the work of several labs over several years. And what we know these ATP-dependent chromatin remodelers do is they take a nucleosome and they can catalyze a variety of different outcomes on this nucleosome. So here's a nucleosome again in cartoon form, and I'm showing here a piece of DNA that is occluded by nucleosome formation. These remodelers can slide the whole histone octamer and make the site accessible. These remodelers can also completely dissociate the histone octamer and free up the piece of DNA. And in some cases, they can exchange pieces of the histone. And so you can take a histone H2, H2B dimer of one type and exchange it with another type. And all of these activities are ATP dependent. These different activities are done by a variety of different chromatin remodeling enzymes that fall into four major classes: the I-switch class, the switch-NIF class, the INO80 class, and the CHD class. I don't expect people to keep the names in mind, but this is just for the folks who study chromatin remodelers to kind of notice that there are at least four different classes, and each class has a slightly different activity and a slightly different mechanism. Now, since these remodelers are making DNA accessible and they're reorganizing nucleosome positions and they're changing how chromatin is assembled on a piece of DNA, it is not surprising that these kinds of machines or remodelers are very important for processes like transcription. And transcription is the context in which most of the activities of these remodelers were originally discovered and well studied. And so I'm showing here a cartoon version of a promoter from budding yeast, where you have nucleosomes positioned around the transcription start site, creating a nucleosome depleted region. And you have a variety of remodelers coming in and acting on nucleosomes to make this transcription start site accessible, and also to kind of reorganize the nucleosome landscape as RNA polymerase moves through a nucleosome. So as you can imagine, if RNA polymerase moves through a nucleosome to transcribe RNA, it's going to be quite disruptive. It's going to move nucleosomes around. It's going to break nucleosomes apart. And part of the job of these remodeling complexes has been shown to be to reset the nucleosome architecture so that you can prevent cryptic transcription and allow for proper transcription from the right transcription start site. Now, what is becoming clear, or has become clear over the last 5 years or so, is that as RNA polymerase goes through a nucleosome, it creates these subnucleosomal particles called hexasomes. And what you have in a hexasome is a nucleosome that has lost an H2H2B dimer. And this has raised a question about whether these hexasomes are simply collateral damage because RNA polymerase is going through and it's creating these broken nucleosomes, or whether they also play a regulatory role. Now, there's a whole class of proteins called histone chaperones that I'm not going to talk about, but that many folks have shown can restore these broken nucleosomes by adding in the H2H2B dimer that's fallen apart. Okay? So there is a process in place in the cell for making hexasomes back into nucleosomes. But the work of several folks shown here, Heinekoff, Straditsky, and Frank Pugh, have shown that these kinds of subnucleosomal particles hang out for a while in the context of transcription. And so that brings back the question, do they actually play a regulatory role? Now, there are a few different ways to address this. And the approach that we are taking is to ask, if these hexasomes matter, if they play a role, do any of the chromatin remodeling enzymes that we originally thought of as acting on nucleosomes, do they also act on hexasomes? And if these enzymes can act on hexasomes, that perhaps there's a regulatory role for hexasomes. So what do we know about chromatin remodelers that slide nucleosomes? Okay. So three out of the four remodelers that I talked about, the CHT class, the I-switch class, and the INO80 class, are known to predominantly slide nucleosomes and not do other kind of disruptive actions on nucleosomes. Greg Bauman's lab has shown that the CHT class of remodelers in yeast can slide hexasomes, it can move hexasomes, and it does so with roughly similar efficiencies as sliding a nucleosome. We've shown in data that I won't share today for lack of time, the I-switch class cannot slide hexasomes. So it cannot move these broken particles. And so what we were curious about was whether the third class of chromatin remodeling machines, INO80, which slides nucleosomes, can slide hexasomes. And if you can learn something about the role of hexasomes through studying the impact of INO80 on hexasomes. And the other reason for being curious about INO80 and its role on hexasome regulation is that INO80 plays major roles not just in transcription, but also in DNA replication and DNA repair, two other contexts where you're likely to see subnucleosomal particles as new histones are being deposited. And again, histones are being dissociated in the context of DNA repair processes. And so here on the right is a cartoon version of INO80. It has many subunits. The whole complex is called INO80 in caps, and the core ATPase subunit of this complex is called INO80 in small letters, just something to keep in mind. It has a variety of the other subunits that I will refer to as we go through the talk. Okay. So this is how INO80 looks in terms of the cartoon form. And what we were curious about was whether INO80 can actually slide hexasomes. Okay. And so I need to give you a little bit of context for the experiments we did in the context of hexasome sliding. Here is how the nucleosome architecture looks in the gene bodies of yeast. Between nucleosomes where transcription starts, there's a large amount of DNA, which is greater than 80 base pairs. But between nucleosomes in the gene body, nucleosomes are very closely positioned on the order of 20 base pairs apart. And this is relevant because INO80 has been shown to have an impact on elongation of transcription. So it's known to have some effect in gene bodies. But in previous work, what we had shown is that if you give INO80 a nucleosome with varying lengths of DNA and you shorten the length of DNA to 40 base pairs, its activity as measured by a Fred-based assay decreases by 100-fold. So INO80, which can normally slide nucleosomes with long pieces of DNA greater than 80 base pairs on the order of minutes, if you shorten this flanking DNA, it now slides nucleosomes 100-fold more slowly. So that raised this question about if INO80 is actually acting on nucleosomes in gene bodies, how is it sliding nucleosomes when the extra length of DNA coming out from a nucleosome is much, much shorter than what INO80 is capable of using to slide nucleosomes fast. Okay? And so this is the context for the biochemistry I'm about to show you. And so what we wanted to ask was, does INO80 directly move hexasomes? Okay. And this experiment was done in the context of a bigger question that was asked by two former folks from the lab, Laura Shea, a former postdoctoral fellow in the lab, and Miriam Gourdet, a former graduate student. They were more broadly interested in understanding if INO80 regulates hexasomes. And so this is a part of the data from their paper, which was published a few years ago. So what they did was they said, let's compare the rate at which N080 moves nucleosomes to the rate at which N080 moves hexasomes. So to do this, they used a classic assay to look at nucleosome sliding. What this assay takes advantage of is that if you have a nucleosome that's positioned on a short piece of DNA and it's positioned on one end of the DNA, it migrates at a certain position on a native gel. If this nucleosome is moved away from the end towards the center, its migration on a native gel changes. It migrates much more slowly. Okay? And N080 has been shown to move nucleosomes from the end of a short piece of DNA to the center. Okay? And this is the activity that is sensitive to the length of the flanking DNA. So here's an experiment where you have a nucleosome with 40 base pairs of DNA, and you add N080 to it, and you look at how fast it moves this nucleosome from the end of the DNA to the center. And as you can see, it is really slow. It's on the order of hours. This time scale is not really compatible with transcription that happens on the order of minutes or faster. So what happens if you take off a histone H2H2B dimer and ask now what happens to the sliding rate by N080? And that's the experiment Laura and Miriam did. If you do that, if you create a hexasome, and you can create different types of hexasomes, you can create a hexasome with even shorter flanking DNA to mimic what happens in the gene bodies of yeast, or you can keep the DNA 40 base pairs. In either case, when you remove an H2H2B dimer, now the rate at which N080 slides hexasomes dramatically increases. And you can quantify this to get rate constants. And what Laura and Miriam found was that the rate constant for sliding hexasomes was 60-fold faster than sliding the corresponding nucleosome. So this is not a 2-fold effect or a 5-fold effect, but a 60-fold effect. So this was pretty exciting because this was, for us, the first example of a chromatin remodeling enzyme that preferred hexasomes. And it suggested that hexasomes may not just be collateral damage if you have a whole machinery dedicated to moving them relative to nucleosomes. Now, this was exciting. It said N080 preferred hexasomes by a large amount, but it raised another question. We know that N080 can act on nucleosomes, and we've just seen that N080 can move hexasomes much faster. But hexasomes look very different than nucleosomes. And even in this simple cartoon form, you can see that the path of the DNA wrapped around a hexasome is very different than the path of the DNA wrapped around a nucleosome. Okay? In addition, and this is in addition to missing a whole component of a nucleosome, the H2H2B dimer. Now, when we learn about enzymes in grad school or undergrad, we know that enzymes have active sites that recognize specific substrates. And they're best suited to acting on substrates that can be best kind of acted on that match the active site of an enzyme. So how do we imagine the corresponding active site of N080 can accommodate both a nucleosome and a hexasome? How is it that N080 can act on both substrates? And why is it better on a hexasome? And so to get at this answer, we did a variety of experiments, but one of the experiments that was most clear was when we got a structure of N080 bound to a hexasome. And this question was asked by these wonderful folks, Miriam Gourdet, the former graduate student who also did work on the hexasome sliding. Elise Munoz, also a former graduate student. Sorry, my Zoom is not behaving. Here we go. And the two of them collaborated with Howeve, a very talented postdoctoral fellow in my colleague Yifan Cheng's lab, to see whether they could get a cryo-EM structure of N080 bound to a hexasome. This study was published last year. So I'll just share some of the highlights of this study. Now, before I share the structure that they obtained on hexasomes, I need to give you a little bit of context and background for what is known about the structures of other remodelers on nucleosomes. So here again is the cartoon version of a nucleosome. And there are standard ways of assigning DNA locations on a nucleosome. And so the center of the DNA on a nucleosome, called the dyad, is called a superhelical location 0. And every 10 base pairs, the minor groove or corresponding the major groove faces out. So you can have kind of waypoints on nucleosomal DNA that are marked. And this is largely so you can compare how different structures of chromatin regulators match up with different types of nucleosomes and different types of regulators. So 20 base pairs out from the center of the nucleosome would be SHL -2. This side would be SHL +2. Okay? Why am I telling you this level of gory detail about how nucleosomes are kind of, how DNA positions are assigned? I'm telling you this because INO80 is kind of weird, okay? And not weird in the sense we're hearing about in the political world, weird in a different way. The problem with a Zoom talk is I can't hear if people are laughing. So you kind of have to pardon me. My jokes might fall flat. So what we know about most chromatin remodelers is that if you look at their cryo-EM structures, their ATPase subunits sit at this location on the nucleosome. This includes the I-switch ATPase and the switch 2 SNF2 ATPase. This also includes the ATPase subunit of SWR, which is in the same class as INO80. Okay? So the ATPase subunits of these complexes sit at SHL -2. INO80 is the only remodeling complex whose ATPase subunit sits at SHL -6. Okay? So this has been a mystery, and many folks, including us, have kind of hypothesized that maybe this says that INO80's action on nucleosomes is very different than the action of all of these other chromatin remodelers. So this is the context in which I'm now going to share with you the structure on hexasomes. So first, this is the structure we obtained with INO80 purified from Cerevisiae or budding yeast on a nucleosome. And I also want to mention that the first structure of INO80, of a yeast INO80 on nucleosomes was obtained by the Usterman lab in the Hofner lab. And we just reproduced this structure with Cerevisiae INO80. And we got the same structure as Usterman and Hofner got in 2018. So this is very similar to a published structure for more than five years ago. And what you can see in this structure is that the ATPase subunit of INO80, shown here in red, binds at SHL -6, okay, on the edge of the nucleosome. There's another subunit here, ARP5, that holds on to another region of the nucleosome. So you can see that INO80 is actually holding the nucleosome quite tightly in at least two locations. There are other subunits of INO80 I'm not showing you for the sake of clarity. Okay. So here's how INO80 looks on the context of a nucleosome. What happens on a hexasome? In the same orientation, if you now look at our structure on a hexasome, now the INO80 ATPase has switched to SHL -2. Okay? So now the whole complex has rotated, and it is sitting so that the ATPase of INO80 now sits where the ATPase of all other remodelers sit on a nucleosome. Okay? So this was pretty exciting because this said that INO80 may not be that weird. In the context of a hexasome, its ATPase subunit is sitting in the same location as other remodelers sit on a nucleosome. And just to give you a sense of how much of a dramatic change this is, I'm going to play a movie comparing the locations of INO80 on a nucleosome and a hexasome. So we started with a nucleosome, and now you're seeing the dimer lost and how much of a rotation you need to have to get the INO80 ATPase to its location on a hexasome. Okay. So this brought up another question. It said, okay, INO80 may slide hexasomes by placing its ATPase subunit at SHL -2. But what about nucleosomes? Is it sliding nucleosomes by putting its ATPase subunit at SHL -6, or does this ATPase subunit actually move to SHL -2 even in the context of nucleosomes? So how can we address this question? How can we address whether even on nucleosomes, this ATPase subunit has to make its way here before it can slide a nucleosome? Ideally, it would be great if we could trap a structure of INO80 in action on a nucleosome, but we are not there yet. So while we are trying to get these structures, can we do some biochemistry? And to do this biochemistry, we turn to a really, really old approach, a very classic approach that originated from studies of DNA helicases. And that's the approach of putting gaps in double-stranded DNA. And what several folks shown here in this list here have done with chromatin remodelers is to show that if you put a gap in DNA that's wrapped around a nucleosome, that can tell you whether a remodeling ATPase is acting at that location or not. So if you put a gap at a location where the remodeling ATPase is acting, that greatly inhibits the ability of the remodeler to slide the DNA across the histone octamer. So having a gap there inhibits or slows down the activity of the remodeler in moving the DNA across the histone octamer. So we said, can we use this gap method to ask where on the nucleosome do gaps inhibit nucleosome sliding, and where on a hexasome do gaps inhibit hexasome sliding by INO80? And these gap nucleosomes were made by Elise in the lab with a lot of guidance and help from Sebastian Diendl, who was very generous in sharing their protocols with us to make these substrates. And so I'm going to now show you the data that Elise got with these gap substrates. So again, the idea was that if the ATPase subunit of INO80 is acting from SHL-6, putting a gap here would inhibit nucleosome sliding. If it had to get to SHL-2, then putting a gap at SHL-2 would inhibit nucleosome sliding. And correspondingly, if it was acting on hexasomes from SHL-2, putting a gap at SHL-2 would inhibit hexasome sliding. Okay. So what did Elise find? I'll first show you what she found with hexasomes. So here's a hexasome with no gaps. And if you look at its sliding using our native gel assay, it slides hexasomes pretty rapidly. If you put a gap at SHL-2, INO80's ability to slide hexasomes dramatically decreases. It goes down by more than 100-fold, consistent with what we see in the structures that the ATPase is sitting here. What happens if you put a gap at -6 in the context of hexasomes? You don't really see any inhibition of sliding, and within error is the same as a wild type, a hexasome with no gaps. Okay? And that's consistent with the structures, again, where we don't see any real interactions of INO80 at SHL-6. Okay. What happens in the context of nucleosomes? Here's a nucleosome with no gaps and with a long piece of DNA now, so INO80 can actually slide it fast. And what we find is that if you put a gap at SHL-6, you dramatically decrease the rate of sliding, again, about 100-fold at least. And so this is consistent with the structures where you see the ATPase sitting here at SHL-6. But what happens if you put a gap at SHL-2? If you put a gap at SHL-2, you also inhibit nucleosome sliding. And so you again get at least 100-fold decrease in how quickly INO80 slides nucleosomes. So this was kind of surprising because this said that something about INO80 acting on nucleosomes made this location and this location important for the ATPase. And so I'll come back to our working model for what is happening on nucleosomes with INO80. What we propose is that this is the ground state of INO80 on nucleosomes, where INO80 is bound so that its ATPase subunit is sitting at SHL-6, and the ARP5 subunit, which also grabs onto another part of the nucleosome, is sitting near SHL-2. But what we propose is for INO80 to get into an active conformation that can slide the DNA across the histone octamer, it has to rotate around the nucleosome so that the ATPase subunit now lands up at SHL-2. And it is from this location that it can slide DNA across the histone octamer. And so in this model, the rate limiting step for sliding nucleosomes is this rotation around a nucleosome. And the gap at SHL-6, we propose, inhibits the rotation, the beginning of the rotation of INO80 across the histone octamer. And so this, to us, also explains why sliding on nucleosomes is slower than sliding on hexasomes, because in the context of nucleosomes, we propose there's this additional rate limiting step that can be highly regulatory in the context of INO80's action in a cell. So then why is it better with hexasomes? With hexasomes, it's more straightforward. Our structures and our biochemistry indicate that the INO80 ATPase starts out in its ground state by sitting at SHL-2. And from here, it can just go because this is the active conformation for all remodelers on nucleosomes, and it can slide DNA. And if you compare the two, again, nucleosome sliding we propose is slower because it has this extra rate limiting step. So this, to us, was exciting because it said this machine was quite sophisticated. It preferred hexasomes, and its ability to slide nucleosomes was very highly regulated. So coming back to why is this important now? We started with, were subnucleosomal particles collateral damage, or do they actually have a role? Broadly speaking, our observation that INO80 prefers to bind hexasomes and binds hexasomes in a more active conformation than it binds nucleosomes suggests that hexasomes must be important, that you have a whole remodeling complex that seemingly prefers hexasomes means that hexasomes must play a regulatory role. Okay? So what could that regulatory role be? The clues have always been there in the literature, but we can come back to these clues about what hexasomes might be doing in a cell. So first, coming back to INO80, the hexasome preference of INO80 might actually explain INO80's ability to participate in multiple disruptive processes, not just transcription, but DNA replication and DNA repair, where these kinds of subnucleosomal particles have been known to be present in large quantities. The restoration of subnucleosomal particles may also inhibit cryptic transcription. What's known is that when RNA polymerase makes these hexasomes in the context of transcription, it also moves those particles off base, literally off base, to expose cryptic transcription sites. So the ability of INO80 to restore the positions of hexasomes might inhibit cryptic transcription. What's also known from really pioneering work from Vasily Studitsky's lab is that subnucleosomal particles create a much more permissive transcription environment. So you can imagine one round of transcription creating these hexasomes, and next rounds of transcription now have an easier time moving through these subnucleosomal particles. And INO80 may play a role then in maintaining and sliding these subnucleosomal particles in the context of this kind of permissive environment. From the context of biophysics and chromatin folding, what is kind of interesting and really fun to think about is that when you form a hexasome, you don't just lose a piece of the histone, you also dramatically change the path of the DNA. And what we are curious about is how dramatically changing this path of DNA can affect higher order chromatin folding. And so you can imagine in condensed chromatin, if you just lose a few dimers from a histone octamer, you could in principle change how the rest of the chromatin is folding through cooperative effects. So that's just speculation. And finally, what our studies with INO80 are telling us that its ability to slide nucleosomes is highly regulated. It's highly regulated in terms of the flanking DNA and whether or not you have a dimer or not. And so we are currently doing studies on understanding why the flanking DNA affects INO80's abilities to slide nucleosomes so much. So stay tuned. Those studies are forthcoming. So now I'll switch to the second part, which is trying to understand how ATP-dependent chromatin remodeling can happen in crowded chromatin. And this study has been led by a very talented graduate student in the lab, Camille Moore, who's a joint graduate student between my lab and the lab of my colleague, Vijay Ramani, who I will bring up again as I show more data. So the question that Camille was curious about is that we know a lot about how chromatin remodelers act on nucleosomes through studies, the kinds of studies I just showed you, in solution, in dilute solutions where we do really careful biochemistry and structure. And how can we relate those studies to understanding how chromatin remodelers act in a much more crowded environment? So that was her broader question. And the question also arose from the kind of this understanding that we know a lot about what remodelers do to nucleosomes, something I introduced in the beginning. And we know that there are many different kinds of remodeling complexes. And what is clear is that of all these remodeling complexes from the switch-SNF family are the most versatile because they can do all of these different activities on a nucleosome. They can slide nucleosomes, they can completely disrupt a histone octamer, and they can exchange pieces of histone. And so Camille was interested in understanding how the switch-SNF class of remodelers might act in a crowded chromatin context. And to do this, she chose to study the budding yeast, a major switch-SNF complex in budding yeast, which is RISC. And what we know is that RISC catalyzes all of these chromatin rearrangements, nucleosome rearrangements. Shown on the left is a cryo-EM structure from Zhu Chen Chen's lab, and there are many other cryo-EM structures of RISC bound to a nucleosome. And what I want to emphasize is that many other chromatin remodelers, RISC is part of a large multi-subunit complex. And I've highlighted only here the ATPase subunit of RISC, which also sits at SHL2, like the other remodelers I talked about. Okay. So the main point I want to get across here is that RISC is a large multi-subunit complex, and it covers a large area of the nucleosome when it binds a nucleosome. Okay. What's known from the work of many people is that RISC plays critical roles in the activation of transcription. And it is also linked to decondensation of chromatin during transcription. Okay. And so the question was, can we do biochemistry in crowded chromatin to really link its activity on nucleosomes to its activity in the context of crowded chromatin? So how could we do that? Well, what is quite fortunate is work from the Rosen lab a few years ago had shown that if you take chromatin arrays, if you take long beads of nucleosomes on long pieces of DNA and put them together in physiological buffer conditions, you can create a chromatin condensates, where here they have labeled histone H2A, and here they have labeled DNA, where within these condensates, the concentrations of nucleosomes are on the order of hundreds of micromolar. Okay? And these concentrations of nucleosomes in these in vitro reconstituted condensates are very close to the concentrations of nucleosomes predicted to occur in a nucleus. Okay? And so now we have a system where we can recreate the crowded conditions in the nucleus in a test tube and study how nucleosomes in this crowded context can be acted on by chromatin remodeling enzymes. Okay, so that's great. We have a system to recreate the crowded environment in a test tube. How do we look at what's happening to these nucleosomes in the context of remodeling? And here's where Vijay Ramani is really a powerful technique of mapping nucleosomes at single molecule resolution comes in. So what Vijay and his lab have pioneered is this approach called SAMOSA. It's a single molecule approach to map the positions of individual nucleosomes on long fibers of chromatin. Okay. And so this approach takes advantage of the fact that if you have a nucleosome sitting on a long piece of DNA, the DNA occluded by the nucleosome is less accessible to adenosine methylase, such as EcoG2. So you can literally footprint nucleosome positions, and then you can read out the footprints by long-read sequencing, packed by long-read sequencing, because the regions that are occluded by histone octamers are not methylated. And when you do this, you can get really nice footprints of nucleosomes on long pieces of DNA. So what I'm showing you here are individual molecules that have been mapped, all aligned and stacked. And these are assembled on a native piece of DNA from a defined gene in a mouse genome, and about 3.5 kb long. And you can see that the regions that are inaccessible are mapped to the footprints that you would expect for a nucleosome. And when you align them, you can see stripes of nucleosome footprints, and you can see regions that are accessible where you don't have nucleosomes sitting, or we don't have histone octamers sitting. And you can do this as a function of varying histone octamers. You can get different nucleosome densities. And then correspondingly, you can see what the propensity is of these chromatin arrays to form condensates, or these phase-separated droplets. Okay? And so by using this method, what Camille was able to do was to simultaneously visualize, at the same time visualize condensates while also prepping the chromatin that had been acted on by chromatin remodelers to assess whether they had actually moved these nucleosomes around. Okay. So what I'm going to show you next is some of Camille's data where she added RISC and chromatin together and asked, when you mix RISC and chromatin together, what happens to the condensates, and can RISC actually remodel nucleosomes in these condensates? Okay. And so what I'm showing you on the right is what happens when you just have buffer and ATP. And you can see, again, the stripes of occlusion where nucleosomes are sitting, very well-defined locations. Again, each of these lines is a single fiber. When you add ATP, it's very clear that these locations become fuzzy. Nucleosomes have been moved around. They have been disrupted. And Camille has a lot of beautiful quantification of this data showing that in most cases, nucleosomes have been moved around. In very few cases, you get histone eviction. So mostly, you're moving nucleosomes around and you're redistributing nucleosomes on these long fibers of chromatin. Okay? So this is what happens with ATP. What happens when you add ATP analogs? When you add ADP, the locations of the nucleosomes don't change, they become slightly fuzzy. And we think this is because when RISC binds nucleosomes in the ADP state, it's locally destabilizing perhaps the nucleosome footprint. When you add ANP-PNP, non-hydrolyzable ATP, nothing really changes, okay? So well-controlled experiment, the effects we are seeing are dependent on ATP hydrolysis. What happens to the corresponding condensates? Okay. This is where we got a little bit of a surprise. Here's how the condensates look. And I forgot to mention, we visualize these condensates because we have end-labeled the DNA with a fluorescent probe. So each of these is visualized using fluorescence microscopy, confocal fluorescence microscopy. When you don't have RISC, you have condensates. When you have RISC and ATP, the condensates get a little smaller and a little dimmer, and we'll come back to this in a minute. But what was surprising was when you have ADP or ANP-PNP, now the condensates dissolve. Okay. So this was actually the opposite of what we had been predicting. We had been initially predicting that RISC and other chromatin remodelers might act as molecular stir bars. They might use the energy of ATP to get inside condensed chromatin and dissolve the condensates. But instead, what we were finding is that you needed ATP to maintain the condensates if we just took this data at face value. But if you put RISC either in a single ATP state, ADP state, or ANP-PNP state, now the condensates fell apart. So what could be going on? To get some more insight into this observation, Camille decided to do the experiment slightly differently. She decided to first make the condensates separately and then add in RISC and ask what happens when RISC is added to the preformed condensates, okay? And then follow the process, look at whether you get remodeling, and look at what happens to the condensates themselves. Okay. So here's what she sees. If you have the condensates, you preform the condensates, and now you add in RISC, what she noticed was the condensates get a little bit larger and they get less intense. Okay. So what this was telling us is that RISC is decompacting the condensates. And you can quantify this as a function of different ATP concentrations. And consistently, when you add RISC, you decompact the condensates. So this was exciting because this is the first biochemical evidence that RISC, which has not been indicated to do this in cells, is actually directly decondensing condensed chromatin. But as she was doing this, she was also taking movies, and she noticed something more interesting. She noticed that when she took movies to look at what was happening, adding RISC increased the dynamics and dynamics of the droplets. Not only did it make the droplets less dense, but the droplets now started showing local movements. And I want to just let people know this is a video that is looping. Okay, so it's the same loop that is playing again and again. So this was pretty surprising to us and made us think about what might be going on. And Camille did a bunch of controls. She asked what happens if she increases ATP, and the movement increases as a function of ATP concentration. Okay? And so this was very curious, and it was also very exciting because it kind of forced us to step back and think very systematically about why RISC might be doing this, and how could we take everything we know about RISC at the level of a nucleosome and use that to understand what was happening in the context of these condensates. And so this is one of the models that we are working with. We know, as I showed you earlier, that RISC, when it binds a nucleosome, occludes large surfaces of the nucleosome. And so in this model, what we hypothesize is that as RISC cycles through the ATPase cycle, like any other motor protein would do or any other machine would do, it will go through an ATP state, an activated or hydrolyzed ATP state, an ADP state, and an APO state. Okay? What we propose is in all states, except this activated state, where ATP is on the verge of being hydrolyzed or has just been hydrolyzed, RISC binds nucleosomes and occludes the nucleosomal surface. And when it does so, internucleosomal contracts are broken. Because what maintains condensates, what maintains chromatin compaction, it's contacts between two nucleosomes. If RISC binds these nucleosomes and prevents the nucleosomes from interacting with other nucleosomes by occluding those interfaces, then you're going to have local breakage of internucleosomal context. So you're going to have local decompaction. Okay? But in the ATPase cycle, there's going to be some points where RISC lets go. And that's when you can form internucleosomal contacts. Okay? And the time scales of these different processes is going to determine how often internucleosomal contacts are formed and how often they're broken. And what we are proposing is that because this is happening in an asynchronous manner all across the condensates, you land up getting local areas where things are broken and formed, leading to very local movements in condensates. So this is not any kind of directional movement because this is happening in an asynchronous manner. But what was exciting to us was it actually helps us begin to link action at the nucleosome scale to action at a condensate scale. And what this might do is might cause transient changes in local viscosity. Now, this brings up the question of, okay, we've added in RISC. What's the stoichiometry of RISC in these condensates? And so Camille did a different kind of experiment. She fluorescently labeled the RISC in addition to labeling the DNA. And she asked what happens when RISC gets inside these condensates. And she got two types of data from her experiments. The first kind of data that she got was she could now quantify the ratio of RISC to nucleosomes and get a stoichiometry. And what she found was even if she added stoichiometric RISC to the condensates, there was not enough room to accommodate stoichiometric RISC in the condensates. And she still got substoichiometric RISC getting inside the condensate. So inside the condensates, there was the most RISC she could get was 1 RISC per 13 nucleosomes. Okay. So despite being substoichiometric, RISC was able to cause these dynamic motions inside the condensates. And we are beginning to get some idea as to what that might happen through FRAP studies. So if you FRAP RISC in the presence of ATP within these condensates, what Camille found is that RISC can diffuse on the minutes time scale across these micron scale distances. Okay, so its diffusion can happen. It happens on the minutes time scale. So in principle, RISC can diffuse through the condensate and somehow through a cooperative effect by maybe disrupting just a few internucleosomal interactions, create these internal dynamics that manifest as movements, local movements in the condensates. Okay. Clearly a lot to still be figured out, but this is where we are. But we are excited because we are now beginning to really do biochemistry in condensed chromatin and discover new phenomena at that scale. And so I'll end with some speculation and biological implications. We think that our data for the first time shows that RISC remodeling can decompact chromatin states independent of transcription. What might this do in the context of nucleosomes in a cell? We think RISC might actually enable increased mixing within chromatin condensates. So this might be its additional function in addition to moving nucleosomes around or kicking histones off. What we are finding is evidence that remodelers can drive micron scale motions of condensed chromatin, and perhaps this is very relevant to other processes in the cell at the chromosome scale. And at a much more kind of philosophical level, when remodelers were initially discovered, a lot of genetics showed that they were important because you mutate the gene and you get cell death, or you get defects in differentiation, or you get embryonic lethality. And the function of a remodeler was assumed to be based on biochemical assays, ability to move nucleosomes around or to kick histones off. But our assessment of function is limited by the assays we can use. And so what we'd like to propose is that the biological importance of remodelers may not just reflect their effect on nucleosomes, but also their corresponding effects on large-scale chromosome dynamics or chromatin dynamics. Okay. With that, I'll end. Thank you for coming to this seminar. And I want to thank all of these wonderful folks. For the first part of the story, Laura and Miriam got the hexasome story started. Elise and Upneet, and I didn't get a chance to talk about Upneet's work on flanking DNA length dependence. Elise, Upneet, and how really contributed to the InnoAD story. And the InnoAD story would not have happened without the work of Coral Zhu, who was the first student in the lab who actually started the InnoAD project. And that's why we are where we are now. and Coral was helped by Nathan and Stephanie in the early days. Also want to thank Camille for taking on this challenging project of looking at remodeling in condensates, and my two wonderful collaborators, Vijay Ramani and Yifan Cheng, Sebastian and Greg Bauman and his lab for help with correspondingly making gap nucleosomes, and Greg for help with hexasomes. And this is an old photo, new photo is coming, and thank you all.
Thank you so much. That was fantastic. Really appreciate you sharing that with us. We've got a few minutes before the hour, so if there are any questions, feel free to type them in. We've got one right here. All right. Can you see that, Geeta? Do you want to maybe read it out loud before you answer?
Yeah, I see one question from Justice, if I'm getting that right. Thank you, Justice, for your kind remarks. His question is, if you already looked at innovative remodeling at adjacent hexasomes or overlapping nucleosomes, meaning a nucleosome next to a hexasome or even larger arrays, what would we predict to happen? It's a great question. We haven't done that yet, but Vijay and I have been talking. It might be fun to do the samosa experiments with InnoAD on arrays and see what happens when we have hexasomes on it. We haven't done it, but it's a good question. We would love to do it and see what we find. So there's a question, shall I take Jason's question or shall I take Ralph or Xiaomin's question? How should I do this, Ben?
I think Xiaomin was maybe just before, so maybe
Okay.
you want to turn off your mic and ask, or
great talk. And honestly, I think Ralph would raise his hand before me, but I will take the honor to ask.
No problem. Just go ahead.
So the second part of talk is fascinating, seeing the RC complex can intermixing the condensates. So I was wondering if you have done an experiment to incorporate histone modification into the system, because RC has so many modules to bind to acylated, methylated histones, and how would those factors influencing the intermixing dynamics?
We actually had also my question. I was specifically wondering about acetylation for risk.
No, great question. We haven't considered it, but I will bring this up with Camille. She's considering it for some other project, but it might be a really nice synergy. What I will say though is that Mike Rosen's lab had shown in their paper using 601 Vitamarase that if you acetylate them, it inhibits condensate formation. And then if you add BRD4, it restores the condensates. And so there's already some data out there showing the effects of acetylation, but it might be interesting to see what happens in the context of risk, because we might have something different. Yeah, absolutely.
Great, thank you.
Thanks, Geeta. Nice talk. Very nice.
Ralph, that's your question. Okay. Yeah.
Yeah, that was also my question. Yeah.
Great.
There's a question from Jason in the chat.
Yep, great. So Jason is asking, do you think there's a connection between the hexasome-specific InnoAD remodeling and the proposed role of InnoAD in H2AZ eviction? Yeah. Let me answer that question. I can say I will speculate. So we haven't, in our experiments, we haven't seen with non-H2AZ nucleosomes, we haven't seen any evidence for eviction in biochemically. So if you take nucleosomes and we do InnoAD remodeling, we don't see the formation of hexasomes. And so it's possible that in a cell, when you have a chaperone, that helps, that you transiently take out an H2H2B dimer, you can slide faster, and somehow that's coupled to putting on something else. That may be a possibility, but on its own, we haven't seen any evidence that it evicts dimers. But it could be, you're muted, Jason.
Sorry, I was thinking maybe if the H2AZ is less strongly associated with the nucleosome, that it might give an opportunity for InnoAD to bind to what is transiently a hexamer, which would have the effect of removing H2AZ, but mainly based on this preference for a hexamer binding.
That's a great question, and Blaine Bartholomew has done a version of that experiment where he showed that with H2AZ containing nucleosomes, N80 slice them about twofold faster.
Mhm.
So and that, I think at least the current interpretation is because the acidic patch is different. But yeah, it may be worth going back to those experiments and seeing whether we see more hexasome formation when we have H2AZ. Yeah.
Thanks. Great talk.
Yeah, thank you.
Okay, Rivan, you want to unmute yourself and ask?
Yes. Hi. Thank you for this really wonderful talk. It's very enjoyable. I have a minor question. So for your Samosa experiment, and when you're quantifying the disruption that RSC does to the nucleosomal arrays, how many samples is that taken over?
When you say, you mean how many repeats?
Yes, how many repeats? Just because when we're thinking about sequencing techniques and quantifying the disruptions there, I'm just sort of curious about how to bridge the two.
So the plot I showed you is one experiment, one reaction, but has multiple molecules in it. Right? And so Camille has done this at least 3 times in 3 experiments and under, maybe slightly different conditions, and she sees very similar results. Does that answer your question?
I think so. Yes. Sorry, I'm just trying to think about number of cells and just sort of when we're thinking about chromatin organization, just the variability among them.
So this story is going to be put on bioRxiv very shortly. So there will be a lot more of the kind of information you're asking there, but in how many times it was done, how the statistics and all of that, because there's a lot of information there also about what fraction of the risk products are dissociated, evicted, what of them are moved, things like that. Yeah.
Great. Thank you. Wonderful talk.
Thank you.
And then one more question in chat.
Yeah. So question from Tom Broker. Linear DNA does not have the winding and twisting limitations of supercoiled DNA. Have you looked at chromatinized supercoiled plasmids? Not yet. And are you talking about for the first part or the second part of the talk, Tom?
Primarily the second part, but either way.
Yeah, we have not. There is another project in the lab where we are looking at changes in linking number. So this may be a good time to revisit this issue. So make closed circular plasmids, make chromatin. Yeah.
As an example, SV40 or papillomavirus DNA within the capsids of the virus particles, if you disrupt the capsid, the viral DNAs remain chromatinized.
Yeah.
So it's a ready source of naturally chromatinized DNA.
Yeah, no, it's a great idea, and it's actually in the old days, Bob Kingston and others, when they were looking at remodeling activity, they would actually look at close circles with nucleosomes, and changes in linking number was a measure of remodeling activity.
Yeah.
Right? So it might be good to come full circle and bring it back to condensates. Yeah.
Okay, and I'll add that our lab has found that if we put in inhibitors of histone deacetylases, replication is blocked. So there's a lot of things that are different between linear DNA and the closed circular forms.
Yeah, no, that's good to know. Thank you.
Thank you.